Satellite cells are muscle stem cells that most people have never heard of, and they're a central part of how creatine actually builds muscle over the long term. Understanding what satellite cells do and how creatine affects them explains one of the most important mechanisms behind the supplement's reputation for supporting muscle growth beyond just phosphocreatine expansion. Every muscle fiber is a long, multinucleated cell. It contains many nuclei along its length. These nuclei control protein synthesis within a limited domain of the fiber, roughly the region of cytoplasm immediately surrounding each nucleus. A muscle fiber that grows substantially larger needs more nuclei to govern the expanded territory of cytoplasm. That's where satellite cells come in. Satellite cells sit dormant just outside muscle fibers, nestled between the fiber membrane and its outer covering. They're true stem cells, capable of dividing and differentiating into mature muscle cells. When a muscle fiber is stressed, by intense exercise, by damage, by mechanical load, signals are released that activate satellite cells. They divide. One daughter cell renews the satellite cell pool, and the other matures and fuses with the muscle fiber, donating its nucleus to the growing fiber. More nuclei means greater protein synthesis capacity, which means the fiber can grow larger and produce more force. This is how muscle tissue accommodates growth beyond a certain threshold. Not just by making more protein in existing nuclei, but by acquiring more nuclei to govern more territory. The number of satellite cells in a muscle and the responsiveness of those satellite cells to activation signals are key factors in long-term muscle growth potential. Creatine supplementation has been shown to enhance satellite cell proliferation independently of exercise, and substantially more so when combined with training. Multiple studies using immunohistochemistry techniques to count satellite cells in muscle biopsy samples have found significantly higher satellite cell numbers in supplemented versus unsupplemented individuals after a training period. The effect is most pronounced in fast-twitch type 2 fibers, the fibers most relevant to strength and power training. In practical terms, creatine supplementation during training doesn't just produce more reps and more volume in the short term. It also drives more nuclear content into your muscle fibers, creating a larger structural capacity for growth that compounds over time. This expanded nuclear content persists even during periods of reduced training. This is one mechanism behind the concept of muscle memory. Muscle that has grown large, acquired extra nuclei, and then undergone atrophy retains those extra nuclei during detraining. When training resumes, those nuclei enable faster regrowth than would be possible in virgin muscle, starting from a smaller nuclear complement. Creatine's enhancement of satellite cell activity during periods of active training means you're not just getting better performance now, you're building a larger nuclear capacity that will serve future training cycles. That's a compounding benefit that the short-term performance data doesn't fully capture. 3 to 5 grams per day, combined with resistance training that genuinely challenges the muscle, is the stimulus that drives the satellite cell response. The magnitude of the effect scales with the intensity of the training. Satellite cells respond proportionally to the signal they receive. The satellite cell response to creatine is also dose-dependent and time-dependent in ways that explain why long-term supplementation produces compounding structural benefits. Short-term creatine supplementation, 4 to 8 weeks diet, produces measurable but modest increases in satellite cell number in training populations. Studies extending to 12 and 24 weeks show larger and more consistent satellite cell accumulation, particularly in individuals performing progressive overload training throughout the period. This is consistent with a model where satellite cell proliferation is a gradual, cumulative process rather than a rapid, acute response. The implication is that the structural benefit of creatine, the nuclear content expansion it drives, builds over the entire duration of supplementation, not just in the first few weeks. Someone who has supplemented creatine consistently for 2 years and trained progressively throughout that period has a different nuclear landscape in their muscle tissue than someone who started 6 months ago. This is one of the less obvious compounding benefits of long-term creatine use that shorter studies can't fully capture. The structural changes creatine enables through satellite cell support are slow, cumulative, and worth investing in for the long term.